Hey everybody. So here are the lecture videos that I promised. I'm sorry that I'm having to do it from my hotel room here at the conference. But you're going to be probably looking at the slides more anyway. So anyway, uh, here is the first of the videos. Uh, the first two videos are going to be about the chemical uh, 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 the chemical senses that we have. And then I will post two about hearing, beginning our discussion of the mechanical. All right, so this video is going to be primarily about the first chemical sense that we're going to discuss, which is olfaction or ear. So let's get going. All right, so first, I want to point out that by chemical senses, what I mean is that these are the two senses that respond to chemicals in your environment. With smell and taste, these are chemicals that you ingest. But you have some other uh, sensations that you can consider chemoreceptive because they are reacting to the presence of chemicals that don't involve you ingesting anything. So, um, you know, when we're talking about the chemical senses, again, we're talking about our perception of chemicals in our environment, and these involve chemically gated receptors. So, we talked about photo gated receptors when we talked about um, vision. Right? So the receptors in your eyes are they're receptive and they're gated. They change their activity due to the presence light, photons. The chemical senses, their receptors change their activity due to the presence of a specific chemical. So, smell, that's olfaction. Um, Taste, that's gustation. And then, like I mentioned earlier, you can talk about certain thermoreceptors being chemoreceptors because um, you have certain free nerve endings that respond to the presence of capsaicin. You probably experience this sometimes if you poured some very hot sauce or accidentally poured some very hot sauce in your hand. You probably notice that the skin kind of feels like it's burning, or that it feels hot when you pour the hot sauce. That's that's a chemically gated free nerve ending in your hand, which is receptive. But uh, almost all of the I'm saying almost all of the chemically gated receptors function in a very similar way, meaning they function specifically they function using a key and lock principle. And so if you think back to the way we talked about chemical communication between neurons in the brain, it's a very similar principle. It's in fact the exact same principle here. So your chemoreceptive receptors only respond to the presence of particular chemicals with a particular molecular structure that then has to attach to a receptor with a matching, or I guess you can kind of think of it like a mirror image, or not mirror image, but the opposite, the lock version for that molecule key. Um, so, you know, if we don't have, so for example, with olfaction, if we don't have a receptor with the appropriate lock, the receptor with the appropriate molecular structure for a chemicals in the environment to attach to, then you can't smell it. So carbon monoxide, we don't have the receptor for carbon monoxide, so we can't smell it. It could fill our houses and kill us, but we can't smell it because we don't have the receptor for it. And this is true of basically all of our chemoreceptors. That if we don't have a receptor for it, we don't experience it. But in all of these situations, when we do encounter the chemical with the appropriate molecular structure to attach to the receptor, when that happens, it triggers a response in the receptor that usually leads to the opening of some kind of channel very often, usually a sodium channel, allowing sodium to rush into the receptor, triggering either altering the responsiveness of the receptor cell or triggering an action. Right. So that's basically true of almost all of the chemical senses. So we're going to start talking about olfaction or sense of smell. And we're going to start talking about the receptors. So your olfactory receptors, or you could just abbreviate it to OR, they tend to be clustered in the back of your nasal cavity, and they're bipolar cells, and they have two processes. One going, I guess you can consider it towards the opening of the nasal cavity, and that process has then these kind of little microcilia, these little, you can think of them like little hairs. And those hairs are embedded in a layer of mucus, all right, inside your nasal cavity. That layer of mucus is the olfactory mucosa. So these microcilia are embedded in the olfactory mucosa. Those microcilia have olfactory receptors on receptor proteins on them. All right, so you've got these microcilia embedded in the mucus of your nasal cavity, and so when odorants, the molecules of chemicals that you smell, enter into your nose, they then come in contact with the receptors on the microcilia in your nasal cavity. Now, the other process from those ORs, from those olfactory receptors, is a outgoing axon that then extends, and you can follow here. So, here's the olfactory receptor, right? And here's that first process with the microcilia extending into the mucus. And then the second process is the axon going back up through this thin layer of porous bone to attach to the to attach to cells in the olfactory bulb. Now, we're going to talk more about the olfactory bulb here in a second, but first, let's focus on those receptor cells. All right, so again, remember that the first process coming from the bipolar olfactory receptor cells goes out, and then it has these microcilia that are embedded in the mucus, and they sit there and wait for odor. Okay? Now, as I said, the receptors the receptor proteins embedded on those microcilia operate on a key and lock principle. And like I said, they're embedded on those microcilia, so I won't belabor that again. But what happens when an odorant comes in and it attaches to those receptors, meaning it has the appropriate molecular structure, is it causes a chain of events. So if you remember back when we were talking about different types of receptors back in the neurotransmission chapter, we talked about signal proteins. And we talked about those signal proteins being attached to G proteins on the inside of the cell. Well, that's what these receptors in the olfactory system that are on the microcilia are. They're signal proteins. So the receptors for olfaction are signal proteins, and they have their receptors on the outside of the cell. When an odorant with the right molecular structure comes in, attaches to the outside receptor of the signal protein, the G protein on the inside becomes active. Specifically, what happens is that a piece of the G protein breaks off. All right. Well, that piece of the G protein activates this, and so that piece of the G protein is a secondary message. Sorry, it activates the secondary message. But that piece of the G protein catalyzes, it activates this adenylyl cyclase, or it's labeled here adenylate cyclase. It activates that. Now, what that does, what that adenylate cyclase does, is it converts ATP into CCAMP or CAMP. All right. Now, CAMP is a I think it's adenosine monophosphate. I'm not certain of that. I don't remember off the top of my head, but uh, it's a chemical that will ultimately open a sodium channel. So you've got your odorant molecule coming in, it attaches to the olfactory receptor, it activates the G protein, meaning a piece of the G protein breaks off. That G protein activates the adenylate cyclase. The adenylate cyclase converts ATP into CAMP, and then CAMP, operating as a secondary messenger, it goes and opens a bunch of sodium channels, allowing sodium then to flood into the cell, causing an action potential. All right. So the whole process, start to finish, odorant comes in, attaches to the receptor, activates the G protein, piece of the G protein breaks off, activates the adenylate cyclase. It converts ATP to CAMP. CAMP opens sodium channel, sodium plugs in, activation. All right. Now, once that action potential occurs, then the signal is sent down the axon. Well, I guess in this picture, I guess you could say it's up the axon. All right, through that fourth bone to the olfactory bulb. All right, and it's these axons then cluster. All right, so nerve fibers are axons, right? These axons cluster into these glom, glom, bleh, glomerul, glomerul. That word. So they cluster into these glomeruli, and these glomeruli are chemotopic, meaning that they each cluster, each glomeruli is uh, is carrying a certain kind information about a certain kind of chemical. And so you can kind of think that the olfactory bulb, which is made up of these glomeruli, is chemotopic, meaning it's organized by the type of chemical being sent. That information in these glomeruli then synapse onto these mitral relay neurons. Now, those mitral relay neurons then synapse into two tracks. So you've got your medial olfactory tract, and you can see this light blue is your medial olfactory tract. And it's medial because, I mean, here's your two lobes, so here's the center of the brain. Remember that medial means towards the middle, so there's your medial olfactory tract. And then you have and the other one is your lateral olfactory tract, all right? Remember, lateral meaning towards the side. So your lateral goes out, medial goes in. Now your medial tract, it goes up all the way to the anterior commissure, and then it crosses over to the contralateral side, whereas the lateral olfactory striae are just their nerve bundles. They're bundles of nerves in a tract. That's all that is. It's guess I forgot that name. Anyway, so they then take them on to their ultimate cortical location. So the olfactory cortex is really, there's not like a single olfactory cortex. It's kind of made up of several. You can think, though, of the piriform cortex as kind of the primary olfactory cortex. But you also have the amygdala and the entorhinal cortex. And so you can see, you've got your tracts that bring them in. Here's the piriform cortex. It then goes on to the amygdala, the entorhinal, and some of it then goes on to the thalamus. Well, that thalamus, specifically the dorsal medial nucleus of the thalamus, then routes the information on to the orbital frontal cortex. And the orbital frontal cortex, you can kind of think of it like an associative cortex. So this is where we start associating, or it's involved in associating memories. And in fact, if you destroy the orbital frontal cortex, then people become incapable of distinguishing between sounds. So there you go. That is your, there you go. That is the olfactory system. So we've talked about how the receptors work. We've talked about once those receptors achieve an exponential, what's the pathway of the olfactory information. And so we've talked about how it gets from the smells in your nose to processing in your brain. Now, I always kind of like to point this out because it's kind of gross, and I'm a dork. But remember that your olfactory system is processing odorants, which are just molecules of the thing you're smelling. So if you smell something, you have molecules of the thing in your nose. So when you're smelling crap, you have crap in your nose. I'm a dork. I think that's hilarious. You may not. But anyway. So anyway, there's the track from reception, activity of the receptors processing in the brain. Now, I want to highlight the orbital frontal cortex role in olfaction because that's going to come back later when we talk about gustation in the next video. So be sure to keep that in mind. All right, guys, that's it for this video. As always, if you have any questions, please send them my way. If not,